Hi, I'm Richard Sever from BioArchive and Cold Spring Harbor Lab. With me I have Ana Maria Cuervo from Albert Einstein College of Medicine. Welcome to Cold Spring Harbor.
Thank you for having me.
And welcome to the Senescence and Aging Symposium here in 2025. You're going to be talking later in the symposium about the role of autophagy in aging. But for those of us who don't remember all the details of autophagy and what it's for, can you just sort of paint a picture of what autophagy is and what its role is in normal physiology?
Yeah, so autophagy is a very fundamental cellular process that takes care of the cleaning your cells basically inside. So normally it's responsible for the degradation of damaged proteins, altered organelles, and this is done by the lysosomes that contain all these enzymes. But more important than the removal of those damaged proteins is the ability to recycle. I know that we all study lysosomes as these garbage containers. I really want to glorify them because I really think these are these very elegant recycling systems. So basically, if you degrade your proteins, you get the amino acids that are not damaged, and you can utilize them for synthesis of new proteins. If you degrade your lipids, you can utilize them for energy. So we all think that in autophagy, the function goes a little beyond quality control and cleaning, and it has important regulatory functions at the level of energetics and also regulating other cellular processes.
And there's like three different types of autophagy?
In the case of mammalian cells, that is what we mostly study, there are three general mechanisms for autophagy that basically just differ of how you take something from the cell and deliver it to a lysosome. So basically you can either wrap it in a double membrane vesicle, and then that one doesn't have enzymes, but if it traffics and fuses with a lysosome, that material is being delivered. So that's called macroautophagy. And when we read in papers, they only say autophagy. 90% of the cases, that's the one. And that's, I mean, it's such an elegant process, and it's conserved from yeast to mammals. And then you have this microautophagy that, imagine you have your lysosome, and then you just have little invaginations of the membrane, so it's taking little bites from the cytosol and then pinch off. So it's called micro because it's a little smaller, but it still can do fractions of organelles. And that one was described originally in yeast, but the genes were not conserved. And about 10 years ago, we identified it in mammals, and instead of happening in lysosomes, it happened in an earlier compartment, late endosomes. So in mammals, it's called endosomal microautophagy, just to differentiate. And then the third one, that is my pet project, that's the one that I discovered, I co-discovered when I was a graduate student. All the credit goes to my mentors. I'm not sure I knew what I was doing, but it was the very first form of autophagy that was shown to be selective, and it's called chaperone-mediated autophagy, or CMA for short. And in that case, the proteins, you don't require invaginations or vesicles. There is a translocation mechanism in the surface of the lysosomes that allows single proteins to be identified, brought to this membrane, and then cross the membrane directly across this translocation mechanism, and then it can be extremely selective. So that was the first one that was described as selective.
And is the selectivity based on the fact that you have a heat shock protein or some other chaperone?
Actually, the selectivity is based on the presence of a targeting motif, so similar to what you have, for example, with ubiquitin proteasome, that you have a degron motif. So here is a pentapeptide that is not a specific sequence, but the property of these 5 amino acids make it to be recognized by a chaperone, and hence the name. So this HSC70 will identify whenever this motif is exposed, or either because the protein unfolds or just because it was part of a complex.
So that's the signal that the protein has gone wrong.
Exactly. So sometimes it's because it's wrong, right? So if it's damaged, you will unfold it, or some conformational change happen, and then you can detect this tag. So the chaperone will just say, okay, this is a protein that has to go through this pathway. Other times are fully functional proteins that are identified to terminate whatever they are doing in the cell. So this kind of confers a lot of regulatory abilities. Imagine if you have enzymes that regulate the glycolysis, and you want to reduce glycolysis. I mean, you can stop transcription, but you still have the enzyme, so it's easier to take them just even they are fully functional. So in that case, there are small conformational changes, not unfolding, but enough for the chaperone to identify this protein has to go. So that confers the selectivity, and then the major difference, because you also use some chaperones for other forms of autophagy, but the major difference is that in this case is direct translocation, and also this pathway, this CMA, appears very late in evolution. It's not present in yeast, not even in flies or C. elegans, so it starts appears in birds and above. So we think that is probably evolutional pressure create this pathway to help a little the proteasome, because in a way it looks more like the proteasome. You have
so
a tag,
fine
very
tuning,
selective, fine-tuning, and single proteins one at a time.
Right, okay. So, I mean, if you're fine-tuning, then it must be a very, very tightly regulated pathway then?
So we are still investigating, even after 30 years, that we've been still investigating. We did relatively recent whole genome CRISPR-I, again, because we didn't have the luxury of yeast and doing those beautiful genetics. We have to wait till mammals we can do RNA interference and CRISPR, right? So we identify, and the mechanisms of regulation, as you guess, are extremely tight. You have regulators at the surface of the lysosome, so only some lysosomes can do this type of autophagy, the CMA, and they are in the surface and they control the binding of the substrates, but also the unfolding, the translocation, so very tightly regulated there, a lot of activity in the surface of the lysosomes, and then you have signaling mechanisms from the nucleus, from the plasma membrane, and even from other organelles. That is pretty cool. We find a peptide that mitochondria can release, and it's a way to tell the lysosomes, get ready because I'm going to make a mess of oxidized protein. So it goes to the lysosome and activate this pathway.
Right. And you mentioned in your abstract that this then starts going wrong as you get old.
Yeah, sadly enough, and this is independent on disease. I mean, our lab study physiological aging and age-related diseases, but even in physiological aging, as you get old, and we have done that in mice, obviously in rodents. We just published a whole atlas of how CMA changes with age and sex in rodents. But also we have done a fair amount of studies now in humans, either samples that we isolate the lysosomes, tissue samples, or just taking advantage of the multiple omics out there. And we have seen that there is this almost universal decrease on CMA activity as we get old. The kinetics are a bit different depending on the organ, and sometimes even inside the organ the cell types, and cell types are more resilient, some of them eventually goes down, and the specific mechanisms are very interesting because they seem to be different in males and females, so that's something we are investigating. But at the end of the day, the major problem that seems to be common is a reduction in this receptor translocation system at the lysosomal membrane as we get old. The reasons are different, so sometimes it's problems with the stability of this receptor in the membrane, sometimes it's transcriptional, sometimes it's reduced translation, depending on the cell type. But the final consequence is you have less of this receptor. You have tons of the chaperone. The chaperone is not a problem, as far as we can tell, because you just use it in a very dynamic manner. If you are not consuming it, it doesn't go inside to be degraded. But this receptor is key, is the limiting step. So we have found that almost in every organ there is a decrease in the total amount of this receptor as we get older.
And because of the nature of the process, this affects so many things downstream then, presumably, like you mentioned metabolism. It's,
So we have been doing systematically, because we wanted to know, as you were pointing out, it's like, okay, what are the consequences? Is this important? I might be very excited about this pathway, but it might not be so important. So what we have done is mimic this aging of this pathway in a young animal, because if we do it in an old animal, so many other things change, right? That is difficult to know. But if we just block this pathway, and we have done it in an organ and cell type specific manner, we have a whole zoo of animals at this moment, but it has been very useful because we have found that, for example, you develop phenotypes of degeneration. So for example, in neurons, if you don't have CMA, it's not that these neurons die right away, but they have this progressive gradual loss of function because of proteotoxicity, and then eventually the animals develop loss of memory, problems with motor coordination. In the eye, also beautiful, very pronounced degenerative phenotype that resembles very much macular degeneration. Skeletal muscle, we have a paper now under revision that also shows this central nucleoli, dystrophy of the muscle. So that's kind of a group of organs. And then we have all these metabolic alterations. So for example, in liver, you develop a fatty liver, you have metabolic syndrome in these animals, and then for example, the adipose tissue, you develop lipid dystrophy, very similar to what has been described with aging or in diabetes. We also see loss of stemness, and we also see problems with circadian rhythms and more malignant transformation. So in many, it's affecting on many levels, because what you say, that the subproteome degraded and regulated by this pathway is kind of broad, but includes things like transcription factors. Imagine some of these amazing transcription factors that we have been hearing during the meeting. If all of a sudden they start accumulating because they cannot be removed timely, you have a problem. You start producing proteins that you shouldn't, right?
Yeah. Well, I guess in the search for kind of like sort of key control regulators that are the causes of aging, it sounds like you're getting very close to having identified one. So does that give you any paths to how you might target?
That's a wonderful question. So, of course, I'm very excited about this pathway, but I don't want to give the impression that loss of CMA is the cause of aging. Of course not. Aging, we know, is a multifactorial, but when you look at these colorful charts of what are the hallmarks or the drivers of aging, the best thing that I think we should get a positive message about that is that, there are 10 or 12, depending on who writes about them. I don't think we have to fix each and every one of them, because they are so interconnected that by fixing a couple of them, you get much more beneficial effect that you're expecting. And I'm saying that because in our case, since we know that this receptor decrease with age, we were like, okay, at least we know something that seems very universal. So we develop a mouse model that basically, we know that till midlife, these animals have good amount of this receptor, and then everything start going downhill. So we didn't want to do a transgenic mouse that has a lot of this receptor when they are born, because that doesn't tell us much about aging. We just did an inducible that whenever the endogenous copy start to go down, we just activate to the same levels that when they were young. So basically, you can compare animals that have not seen a decrease of CMA per life and animals that are wild type and have this decrease. And what we have found, this is still not published, but we hope it will be published soon, these animals live longer, and it's not only, it's 20%, that is pretty good considering mice, and it's not only the maximal lifespan, but also the mean, that is the one that we want to increase, because that means improve health span. So in collaboration with Rafa de Cabo, we have been testing frailty, every kind of parameters. And then, of course, the animals live longer, they are healthier, better memory, better metabolism. And as much as I want to say that that's only for CMA, it's very clear our intervention was only targeting this pathway, because this is unique receptor for this pathway. But then when you look, for example, at mitochondrial function, it's better. You look at the stemness, it's already better. You look at cell-to-cell communication. So I think because you are improving one of these hallmarks of aging, the others are also going to improve, because now they don't have this overload of things that are coming because of malfunction of this pathway. And I think in general, probably we have many angles, like if you remove senescence, the animals also do better, but when you look, they also have better autophagy. So everything is really interconnected.
Yeah. Well, that's great to know, and hopefully that can all be translated to work in humans. It was fascinating talking with you, and good
my
luck
pleasure. Thank you for all these
with the work
questions.
going forward.
Thank you.
I enjoyed chatting with you.
Thank you.